Today we like to start with 1 minute. So today we like to start with another type of organelle present in cell. So once we can begin with the plant cell, just to have an idea once more about another organelle which we are like discuss, which we like to discuss here, chloroplast. So chloroplast is here. Chloroplast we can find inside the cell, and this is your chloroplast. Generally, it is asked, why are plants green? Why are plants green? Generally, we mention that chlorophylls and chloroplasts are there, but our answer must be perfect if we put the basic idea first. These are the certain reasons, but reason must be taken in a different light first. When we talk about this, we actually talk about from the standpoint of the light. So you must take this way only. So light ray falling, the white light ray falling on a leaf, we generally find, or what is found in everywhere, that only the green light wavelength is being reflected, others are absorbed. And the light ray which is coming from the object reaching the eye, and then finally the brain will decide that this is the color of the green. When our eyes are receiving the green light from the object, we get the idea of the color of the object. So remember these things. So as plants are green, because the green wavelength of the light is reflected and not absorbed. This is the reason. This same reason is everywhere. We will also take up another example to understand it. Please write these things. Next, another thing we would like to take up. Another example. So color of the object in front of different colors of light. First, if we take the white light, suppose we are taking the white light. So white light, when it is falling on the leaf of the apple and also on the apple fruit, we get the color of the leaf green, because except the green wavelength, all are absorbed. Green wavelength is reflected. In case of the fruit, red wavelength is reflected, others are absorbed. So we are getting the color of the leaf as green, and the fruit color is red. Suppose if we have a filter, only red light can pass through, then what will be the color of the leaf? At that time, we will not get the color of the leaf as green, because there is no green light wavelength. So we will get a blackish color. But we will certainly have the red color of the fruit, because red light is there. If we take up a filter, only green light can pass through, then we will certainly have the green color of the leaf, but because the reflection is there of the green wavelength, but we will not get the red color of the fruit, because there is absence of red color. There is not, the red color is not being reflected, as it is not there. All the other rest of the colors will be absorbed. Similarly, if we take up only blue light, neither the leaf nor the fruit will show any color, as we are observing in the real nature and other time. So this is the very basic reason behind having the color of any object. So try to put your answer in this light. Now coming to the plastid, one of the important cell organelle present in plants and in some protists. Can you remember what is the definition of plant? Mentioning plants means we are having such type of organisms having eukaryotic nature, multicellular cell walls composed of cellulose. Main pigments are chlorophyll A and B, and also they are autotrophs. Protists are also like that, but some do not have all the features of plants, so they are protists. They are not plants. Like they may be unicellular, they may be they may be having something like chlorophyll A and B are not the main pigments, like phycoerythrin is the main pigment in case of red algae, and carotenoid is the main pigment in case of brown algae. So they belong to protist. Now they also are having the chloroplast. Plastids often contain pigments used in photosynthesis, as we are mentioning, and we know that. And pigments, there are several types of pigment present, and that can reflect different types of light rays, wavelengths. So these are all, so for the color, we generally find this type of things are present in case of, I can also show you a little, I think it is familiar to all of you, when you visit garden, you find multiple colors. So if you are visiting the garden, or sometimes when the insects are visiting different plants, collecting honeys, nectars, so they find this type of things. So when we see the colors, we say we enjoy it and we say so very nice. So these are the types of pigments. We find all of these things. Three types of plastids are present: chromoplast, chloroplast, and leucoplast. I would like to differentiate. I have a type. So types of plastids can be divided into on the basis of the colors or colorless, and there is a special color which is known as green. So if other than green, all should be known as chromoplast. Chrom means color and plast. So from plastid, chloroplast as green, because that is very special. And leucoplast is the type of plastid having no color, but they are reservoir of the different types of food particles. Amyloplast, amylo stands for carbohydrate, elio stands for lipid, and protein, perhaps you know that, so proteinoplast. So amyloplast, elioplast, and proteinoplast, three different types of storage type of plastid, commonly known as leucoplast. They do not have any color pigment which can reflect light and can give us any sensation of light. So chromoplast, chloroplast, leucoplast, and leucoplast can be also divided into three heads: amyloplast, elioplast, and proteinoplast. So this is the very basic different types of plastid which we can obtain in different types of plants and plastids. Now, if you go through the electron microscope pictures and the diagram, the right top corner is the electron microscope view, and the bottom is the right corner is the diagram. There are the stacks of sac-like structure. The sac-like structure is known as thylakoid, and the sac-like structure is known as granum. Plural form is grana. In case of chloroplast form, there are several types of form we can find. The left side, then center, and the right side. Can you identify the three organs? Unicellular organism cannot be plant. So left side, this is Chlamydomonas, and it is unicellular. So it should be under the kingdom Protista, and it should be termed as protist. So Chlamydomonas, then second one Spirogyra, multicellular body, belong to the plant, and this group is the, you can say, the first group of the plants. Then Eulothrix. If you notice the plastid structure, chloroplastic structure, you will find different types are there. First one is cup-shaped. Second, center one is spiral-shaped. Check this one. So shapes are different types, varied types. Cup-shaped, spiral, then girdle-shaped. This is known as girdle-shaped, one after one girdle. So these are girdle-shaped. Also net-like and star-like. I would also like to show you net-like and star-like. So several types of structures may be possible here. Chlamydomonas, please remember, do not forget, it belongs to group protist, not to plants, because they are unicellular. Others, right side and center, are multicellular, so they belong to group plants. Next try to see this one. One is Oedogonium. O is silent. In the name O is silent, Oedogonium, and then your Zygnema. So some star-like and some fiber-like, or so these are this type of. Also, chloroplasts are available in different types of plants and protists. In case of plants, we generally find special type of characteristics. So chloroplast, we would like to start with another feature, try to understand this thing and then to pass. So if we take up one simple leaf as a representation, so if we go inside the leaf, we will find cell. If we go inside the cell and to pick up the chloroplast, then we can study this. So chloroplast is here. Inside, if we go, then we will find thylakoid or vesicle, sac-like structure. And one full stack is known as granum. Inside granum membrane, you will find antenna assembly. Antenna assembly are the several types of pigments which can absorb different wavelengths of light. After receiving different or absorbing wavelengths of light, so they are energized. Finally, all the pigments will send their energy absorbed to a particular type of chlorophyll, or rather chloroplast. You may say chlorophyll better. So that chlorophyll generally release electrons, getting energy. So this chlorophyll becomes positively charged. But the chlorophyll doesn't like to be positively charged all the while. Then how to get the electrons? So who can supply electron to this positively charged chlorophyll? Generally, it is supplied by the water present in the cell. So water gets splitted and giving us the oxygen, molecular oxygen. In case of photosynthesis, we get oxygen from this type of reaction, which is known as photolysis of water. With the help of photon or with the help of sunlight, water gets splitted. And this chlorophyll will get the electron, and hydrogen and half molecule of oxygen are evolved. So this is about the function also, try to understand this one. And so chloroplast is producing this one, and after that, so chloroplast will produce another thing. We will also pass to the next point. So inside that first point is the chloroplasts are all, so chloroplasts absorbed the light rays, and then first splitting of water and production of NADPH. Then next phase, first phase is known as light phase. Next phase is known as dark phase. Though darkness is not required, only light is not required. That is to mention, that's why it is mentioned as either dark phase, better to mention as light independent phase. In this phase, it starts with ribulose bisphosphate. In many exams, this is asked. Ribulose bisphosphate, 5-carbon compound. Generally, this way they are asking, name the first compound required for dark reaction, and this is ribulose bisphosphate, RUBP. When you pass through photosynthesis, I think you can understand it better. So RUBP will accept this carbon dioxide, and then it starts reaction. So Rubisco is one of the enzyme which will combine this or starts the reaction, and there will be formation of 6-carbon compound which is unstable. This is phosphoglyceric acid, then it will produce phosphoglyceraldehyde. So each 6 phosphoglyceraldehyde of molecules, one molecule will come out of the cycle and starts producing glucose. Other 5 will recycle back to produce RUBP. So this is the idea about the photosynthesis. So from one phosphoglyceraldehyde, plant or protist will produce glucose, and afterwards it is converted to sucrose. So now we are talking about the chloroplast. Once back into the chloroplast structure present in case of green plants. These are 2 membrane structure, and that's why this is known as organelle. Membranes are like cell membrane, having two phospholipid layer in which proteins are embedded like iceberg. Remember these things once more. Stroma is the jelly-like substance or the matrix present inside the chloroplast. Thylakoid present and granum and all that parts. Remember the also length and breadth. Generally it is 4 to 10 micrometer, and also the width will be 0.5 to 2 micrometer. How to remember? You should develop in your own way. If you develop this way, I think it will help you. If you draw a picture and on the picture, if you make all the things, then you can still remember it as a photographs. I think it will help you also in exam. And also when you are developing your concept, try to develop the concept. This is very important. So these are the about chloroplasts. One question. Read the question. And I would like to get, so one organelle, if it is missing, then it can create havoc. So most of the photosynthetic organelles will not be able to produce food. Some can do that. Only a few are still left who can do this type of thing because they do not depend for chloroplast. Except few, some like bacteria. So most of the organism will be wiped off from the surface of the earth because they won't get food. There will be no one except few to capture energy and from the inorganic foods, inorganic materials to produce foodstuff, which is required as one mentioned that a food cycle will be stopped, certainly, and liberation of oxygen will be greatly hampered. So on the basis, we are depending on the one organelle here. So this is one important feature. So try to understand. So this one is like this one. We would like to go for another part. What is a pigment? Anyone can define what is pigment? Anyone can define what is pigment? Anyone, quickly. No, I have not seen it. To get the idea, photosynthetic organisms produce specialized molecules which can capture energy, especially from light, and can use this energy into different types of redox reaction in order to form chemical bonds. So in case of glucose, you will find carbon, hydrogen, oxygens all are attached together. How? Because that energy is trapped. So solar energy is trapped and that is formed into the bond energy or the chemical energy. Carbon, oxygen, carbon, hydrogen, carbon, carbon all are attached together in the bond energy. So chemical energy or bond energy, or sometimes it is known as static energy also. So this energy is available because solar energy is there and that is trapped by different pigments. This is very special type of molecules available only in very few types of organisms, especially plants. Just going to that part. So these are the, so this is what is a pigment if you want to remember. So what is pigment? Pigments are the very specialized organelles. These are not organelles, these are molecules, sorry. So they can trap light energy. And if we come to point the chlorophyll structure, just to have a little idea about chlorophyll, chlorophyll having a magnesium inside. Understand this, generally it is asked also. Which element is very essential in case of formation of chlorophyll? Apart from carbon, hydrogen, nitrogen, all this will be required certainly, but there is a very special presence of element magnesium. Similarly, it is in case of hemoglobin, the iron is required. So chlorophyll having magnesium and chlorophyll A having this one group known as methyl. There are several others like chlorophyll B will have aldehyde groups, some others are having different types of. So this is the structure of chlorophyll A, and speciality of this is having magnesium. Carotene, if you want to see carotene and model, so this is carotene. If you like to make projects, you can make this type of things and then to present it and how they are absorbing, how they are releasing electrons, and how they are making the process of photosynthesis properly. So these are the things in case of photosynthesis we find, and also in case of chloroplasts. Generally, we find we change in color in leaves during the fall time. So what is the cause or what is the reason behind it to observe this type of color change? Anyone? So change in color is due to at least two reasons. First one, there is no need of chlorophyll anymore. Why? There is no need of chlorophyll anymore. Why? Fall of colors is actually supported by two things. Chlorophyll pigments are not required anymore because cold climate is, or cold weather is ahead of this time, or cold season is coming. Before fall, so chlorophyll production is stopped. There will be no more photosynthesis during the cold season, so greatly reduced chlorophyll production. And secondly, excretory materials like carotenoids and red, which are red or yellow, are generally visible because of the absence of chlorophylls. And excretory materials are also deposited in the leaves, and leaves get detached because this is the way to remove the excretory products from the plants. Every plant having their different types of removal of excretory product. There is no excretory organ, so they have developed their own way. In some plants, the excretory materials are out of the body with the fall of leaves. So here is one same thing. So fall of leaves can support this type of thing. So fall colors, we find different types of colors as the season progresses towards cold weather or cold season. We find this type of, so there is a great reduction in the chlorophyll production or chloroplast formation. Now, plastid functions, as we have discussed, chloroplasts are responsible for photosynthesis, so for conversion of CO2 to carbohydrates. CO2 to carbohydrates. In this occasion, hydrogen is required, so hydrogen will be supplied from the splitting of the water. As we mentioned in the very early stage, in the light phase, water gets split into hydrogen and oxygen. So hydrogen will be supplied to this carbon dioxide and to produce the carbohydrate. So conversion of carbon dioxide to carbohydrate. Then production of oxygen, a very key function. The atmosphere on the land getting the oxygen mainly from this type of process. And from plants and protists, and many protists are on the sea floor, sea also, on the sea, they are floating in nature, sometimes known as phytoplankton. So production of oxygen is also there. Synthesizing several types of organic materials like amino acids, fatty acids, and lipids. Can anyone say what is lipid and what is the difference between lipid and lipid can be classified into several groups like fats and oils. So those who are liquid in above 20 degrees Celsius or room temperature known as oils, and those who are solid at 20 degrees Celsius, there's those known as solids. Lipids are carbon, hydrogen, oxygen containing compounds where hydrogen and oxygen is not in the ratio of 2:1. So synthesizing this type of things, and another important reduction of nitrite, we generally require nitrogen to be incorporated into organic compounds. Nitrogen is a very important component in body formation of all the living organisms. And how to get it, especially to supply the nitrogen to the plants, we generally supplement with the fertilizer or manures. In that occasion, also we supply the nitrogen. Very few organisms can only absorb the nitrogen from the atmosphere. Most of them cannot use gaseous nitrogen. So introduction of the nitrogen into the organic materials, organic compounds is very important thing. Here it can be done with the help of plastid. So one function of plastid is this. Now we would pass to another cell organelle known as mitochondrion. What is the distinction between mitochondrion and mitochondria? Vijayawada, can you say what is the difference? Mitochondrion, when you are labeling one only, then you have to mention mitochondrion. But when you are labeling many, then you should write mitochondria. Mitochondria known as powerhouse because it releases energy from the foodstuff. As chloroplasts uses solar energy to trap and to produce the foodstuff, and solar energy is utilized for chemical energy. Similarly, mitochondria is releasing that chemical energy for useful energy, and it is enough to produce the ATP. Mitochondria are also bound type of organelles, so these are also known as organelles, or bound type of cell parts, and also known as cell organelles. They have their own DNA and ribosome, so they can have their own protein synthesis, DNA and ribosome. And that's why sometimes it is said, or this is general idea, that mitochondria was earlier one prokaryote and finally inserted, or having been inserted into this another type of cell and a part of the cell, and forming the mitochondria as a part of eukaryotic cell. Maybe in earlier time of the on Earth, it was an independent type of organism. So important for tracing lineage. How to know that human being came from where? Whether it came from North America or from Africa or from Asia or from where? How to understand it? So getting the idea, it can be traced back with the help of the DNA present in mitochondria. Every time mitochondria is given by the mother to the offspring. So DNA can be traced back to the last part of the very ancient generation, and from there you can understand that human beings started from Ethiopia because of this trace, and thought to be once symbiotic bacteria, as we mentioned. So about the mitochondria, you can find all these type of things. Mitochondria, the structure is like this: one simple invagination of the internal membrane or inner membrane. There are two membranes, external and internal. So inner membrane can make several foldings inside. This is known as crista. If it is one, if you say, and if it's more than one, then it will be cristae. So mito is the fibrillous type of granules, and here mitochondria is like this because it is smaller in structure. 3 to 7 micrometer in length, and breadth will be 1 to 1.5 micrometer. Sometimes you require that. What should be the number? It should be the number depending on the energy release required by this cell. In case of the muscle cell, it will be required more because muscle requires energy generation more to work hard. In any other cells, 100 to 300 mitochondria per cell is sufficient to generate energy. But the energy rich condition is required some places, and that time you will find more number of mitochondria are required. In case of the synaptic knob of the neuron, you will find that type of things are there. So functions of mitochondria, I would like to first go with the animation and then to come back to the idea about the functions. So first with the one thing which is known as electron transport. We are talking about the respiration, cellular respiration. Respiration is the process when we are getting the energy from the chemical bonds of the foodstuff. These are of 3 steps. First one, it will be in the cytoplasm. Then next step in the mitochondria. and third step in the Christie. So if we look into the mitochondria in the inner membrane and the external membrane, there is a gaps, the space. The intermembranous space, or in the membrane, we find there are several enzymes or several carriers, especially carriers. NADH, there is a one compound, nicotinamide adenine dinucleotide, and when it is H is there, then it is known as reduced. So NAD is carrying the hydrogen here to the membrane structure. Then this hydrogen will release hydrogen. How this hydrogen is transported? Check it here. So all these are carriers on the membrane. Oxygen is inside here in the matrix. So NADH releasing the hydrogen to the carriers, the hydrogen ion. The electrons are being transported with the carriers, and hydrogen ions are pumped outside the matrix. So they are coming to the intermembranous space. Check this one. So electrons are transported through this carrier. Now coming to this, and hydrogens or protons are pumped into the intermembranous space. And finally, these electrons are reached to one place known as oxygen molecules. In respiration, oxygen molecules are utilized at the very last. They are taking part at the very last. So electrons are being transported through all the carriers, and these are being taken up by the electrons. These electrons are taken up by the oxygen, and then finally joining with the hydrogen will produce water at the end. This is the process of the aerobic respiration, where molecular oxygen is utilized. So this type of things, and now the hydrogen ions or the protons are present here. These are again pumped back and to produce ATP. Check this one, that ATPs are produced. So ATPs are the energy-rich compound which will be used as energy currency. If you go into the market, you can purchase fruits, you can purchase vegetables, you can purchase rice, you can purchase even a mobile phone only by using the rupees, so the money. So here also ATP can be utilized for different types of works, and that's why it is known as energy currency. So energy, ATP is produced here, ATP is produced in this process. So here energy can be produced. So functions of mitochondria, if you want to mention, so two types of stages we can mention here in the aerobic respiration. One is Krebs cycle and terminal pathways, as we shown in the animation. So regulates Krebs cycle and terminal pathways. It is also known as electron transport chain, or in abbreviative form, it is ETC, electron transport chain. So these are the steps generally performed in the mitochondria. If molecular oxygen is available, if it is not, then respiration will not be aerobic in nature. It will be stopped inside the cytoplasm. It will not enter mitochondria. So that's all for today.